The process of fertilization does not end with the entry of the sperm into the egg. Fertilization also leads to the activation of the egg's metabolism, and this is needed in order to initiate the process of development. While the activation of the egg's metabolism is initiated by the fusion of the egg and the sperm, it does not require the fusion of the genetic material from these germ cells. In other words, the fusion of the egg and the sperm pronuclei are not required for the egg's metabolism to be activated. And this is because the events that lead to the activation of the egg's metabolism begin with the binding or the entry of the sperm into the egg. Recall that when the sperm binds to the egg, a calcium wave is initiated at the site of sperm entry. And this increase in calcium concentration is most likely to be mediated by the activation of an enzyme known as phospholipase C. In both mammals and sea urchin, this enzyme is activated as a result of either the binding of the sperm or the entry of the sperm into the egg. This enzyme cleaves a phospholipid at the plasma membrane into diacylglycerol and IP3, and these are second messenger molecules. Remember that second messengers are intracellular molecules that can activate other proteins. IP3 binds to receptors in the endoplasmic reticulum that allow the release of calcium. So the IP3 basically serves to open these calcium channels that are localized in the endoplasmic reticulum. And recall that the calcium that is released from the endoplasmic reticulum is responsible for the cortical granule reaction, and that the calcium occurs as a wave. So this happens as a wave because once a little bit of calcium leaves the endoplasmic reticulum, that same calcium binds to other receptors in the endoplasmic reticulum that open and lead to the release of more calcium. So in addition to these IP3 receptors that are in the ER, there are also calcium trigger calcium release receptors, and that's what leads to the calcium wave. In addition to inducing the cortical granule reaction, which is a slow block to polyspermy, the calcium also leads to the reinitiation of the cell cycle. In other words, these cells are now going to begin to divide, and that's what's needed in order for the embryo to undergo cleavage. But before the fertilized egg can begin to undergo cleavage, obviously the DNA has to be replicated. And this is accomplished indirectly by the increase in calcium concentration, which actually changes the alkalinity of the egg because it activates a sodium proton pump. This sodium proton pump leads to the efflux of protons in exchange for sodium. So that stimulates the synthesis of DNA, the translation of maternal mRNAs, and also cell division. So there are a series of events that occur even before the sperm and the egg pronuclei come together. These events are mediated by the binding or the fusion of the sperm, which leads to an increase in calcium. And they're important for activating the egg's metabolism. Your textbook divides these events into early and late responses, but for simplicity, we're just going to look at these overall. As we've already discussed, calcium release from the endoplasmic reticulum leads to exocytosis of the cortical granules, which is important to prevent polyspermy and to form the hyaline layer. And this is the layer that's going to support the dividing blastomeres. In terms of the activation of the egg's metabolism, this calcium release, along with increases in intracellular pH, are important for stimulating the synthesis of DNA or DNA replication and also protein synthesis. In addition to that, there's also going to be an increase in the amounts of NADP. And NADP is used as an electron carrier in anabolic reactions. In this case, it's needed for the synthesis of membranes. And this is because if the fertilized egg is going to undergo cell divisions, it will need membranes in order to apportion to the dividing cells. Another event that obviously needs to happen is that the cell needs to re-enter the cell cycle. Finally, the cytoplasmic determinants or morphogenetic materials that are deposited in the egg can be localized at this stage by movements of the cytoplasm. It's worth noting that the protein synthesis that is seen early after fertilization is not because of new gene expression, but rather this is protein synthesis that results from the translation of stored maternal mRNAs that were deposited into the oocyte prior to fertilization. Another important event that occurs after fertilization is the fusion of the sperm and the egg pronuclei. Once the sperm fuses with the egg, the mitochondria and the flagellum of the sperm disintegrate. So the mitochondrial genome is transmitted through the maternal line. What the sperm does retain, however, is the centrosome. So in addition to bringing genetic material, the sperm also brings with it a centrosome, which is a microtubule organizing center. That microtubule organizing center will form a network of microtubules, as you can see here, and the sperm and the egg pronuclei will use those microtubules to migrate toward each other. Another event that needs to happen is the replication of the genetic material, because recall that these nuclei are haploid. So in order to divide to produce diploid cells after the first cleavage, that DNA has to be replicated, and that involves the decondensation of the chromatin. The replication can happen at the pronuclear stage prior to the fusion of these pronuclei, or once they fuse to form the zygote nucleus.